our presentation. There is currently intense interest in identifying pathological mediators in the urothelium. Oxidative stress is a fundamental pathological mediator, and ROS-generating enzyme is the only targetable protein. Recently, we have identified such system in the urothelium and also reported its basic mode of actions. The aims of this study are to explore aging-dependent changes in the bladder urothelium using mouse model, and also to identify the presence and function of major Nox subtypes in human urothelial tissue and examine the pathological and physiological roles of such system. We use C57 mice, aging versus young, and also the aging mice on this background, but with Nox1, 2, and 4 knockout. We also used human bladder biopsy during the cystoscopy, and bladder mucosa and smooth muscle were microdissected for physiological measurement. We use the West blot to determine the expression of Nox subtypes and lucigen-enhanced chemiluminescence to quantify Nox NADPH stimulated superoxide production in live tissue, and also we also measured the live tissue ATP release using luciferin luciferase assay. This slide shows the superoxide production is increased in aging urothelium. You can see here, left-hand side, this is the superoxide production in the urothelium. During the aging, this is increased. And on the right-hand side, this is smooth muscle tissue. There's no significant change. So this change is urothelium specific. Now, ROS response can also be changed during the aging to some of the important inflammatory factors. This is an example of response to substance P. On the left-hand side, in the aging group, you can see the mucosa response to the substance P is increased, and it is also increased in the smooth muscle tissue. So in both tissues, the superoxide production is enhanced by the substance P. But this response is not changed in the young group. You can see substance P did not elicit any significant changes in any of the tissue in the young group. However, Nox subtype knockout attenuated ROS upregulation in aging urothelium. You can see the attenuation of such augmentation in each subtype knockout. This white type knockout, Nox1, and this is Nox2, Nox4. So in each subtype knockout, such enhancement is reduced. That means the upregulation of ROS can be attributed to each of the Nox subtypes. Then we explored the Nox protein expression and function in human conditions. You can see there's a clear expression of Nox subtypes 2, 1, and 5 in the human urothelium, and the Nox2 is most predominant. Now, NADPH can significantly stimulate the ROS production, as you can see, significant rise against the basal level. The ROS production can be augmented by important inflammatory factors. This example is angiotensin II. You can see here this is control and application of angiotensin II enhanced superoxide production. The same intervention also augmented ATP release in this tissue. That means such action has important functional relevance. Then we tested another important inflammatory factor, which is also a TBV4 activator, GSK compound. You can see here the superoxide production is enhanced by GSK compound. And again, the same intervention stimulated or augmented ATP release in the same tissue. Now, what about the change of this GSK response during the aging in terms of disease versus control? Now, in the OAB patient, you can see there is a correlation between enhanced ATP release and aging. This is a positive correlation. However, such positive correlation is not observed in the control group. So this specific age-dependent change is particularly important, is relevant to pathology, but not relevant to the normal conditions. From all these results, we conclude that ROS production is upregulated in aging bladder urothelium with contributions from all major Nox subtypes. Nox-ROS system is sensitized by pathologically important inflammatory factors during the aging. In human urothelium, there is the molecular and functional expression of Nox proteins and its major subtype. Such system in human urothelium can be augmented by key inflammatory factors. Such response has pathological influence on urothelium function and also contributes to OAB and aging pathology. Thank you.